In the enzyme catalyst lab, we observed the conversion of hydrogen peroxide to water and oxygen gas by the enzyme catalase. We then measured the amount of oxygen generated and calculated the rate of the enzyme catalyzed reaction. Enzymes catalyze reactions by lowering the activation energy necessary for a reaction to occur. Enzymes are globular proteins. Their folded conformation creates an area known as the active site. Catalysis is an enzyme that is used by almost all aerobic eukaryotes to catalyze the breakdown of hydrogen peroxide, a toxic byproduct of metabolic processes. Every second, catalase can break down 40 million molecules of H2O2 into water and molecular oxygen, which can either be eliminated from the body or used for other cell processes. In this experiment, we investigated the rate at which enzyme catalase converts substrate to product. We allowed the catalase to react with hydrogen peroxide for varying amounts of time and then stopped the reactions by adding H2SO4. Our hypothesis was, if the amount of product synthesized is related to time, then the longer the amount of time the reaction is allowed to occur, the more products will be present because enzymes are not consumed within the reaction and are reusable, so product will accumulate over time.
For this lab, make sure to wear goggles and gloves and wear a safety apron. Since this lab consists of the handling and mixing of acids, wearing safety equipment is a necessity to stay unharmed.
To begin, we first put 10 milliliters of 1.5% concentration of hydrogen peroxide into a plastic cup.
1 milliliter of catalase solution that was kept in the freezer was also put into the cup. After swirling it around, bubbles started to form, which is oxygen from the breakdown of hydrogen peroxide by the catalase.
We started boiling water next and then put a test tube of 5 milliliters of the catalase into the boiling water for 5 minutes. Then we removed the test tube. We got a plastic cup and filled it with the same concentration of hydrogen peroxide and put 1 milliliter of the cooled catalase into the hydrogen peroxide. Once the catalase was added, it didn't boil, which means the enzyme denatured.
For the third part of our lab, we also put 10 milliliters of hydrogen peroxide into 2 separate plastic cups and cut 1 centimeter of liver and potato. We macerated the liver and potato. We then put both into the separate cups of hydrogen peroxide. Both cups started to boil. However, when liver and potato are macerated, then boiled, and then placed into the hydrogen peroxide, we notice that they don't bubble. This is because the enzyme is denatured.
To determine the amount of H2O2 initially present in our 1.5% solution, we need to perform all the steps of the procedure without adding a catalase to the reaction mixture. This amount is known as the baseline. In any series of experiments, a baseline should be established first. This is what we did in Part B. For Part B of our lab, we placed 10 milliliters of 1.5% H2O2 into a clean glass beaker. We then added 1 milliliter of H2O into our H2O2 solution. Then we added 10 milliliters of H2SO4 using extreme care. It's important to use extreme care in handling acid. Your teachers should instruct you the proper safety procedures for handling such hazardous materials. We mixed the solution together well. We removed 5 milliliters of this mixture and put it into its own separate beaker. Next, we placed the beaker containing the sample over a white piece of paper. We used a burette or a 5 milliliter pipette to add KMNO4 a drop at a time to the solution until a persistent pink or brown color was obtained. We gently swirled the solution after adding each drop. We made sure that we understood the calibrations on the burette or syringe. This process did take a couple minutes, so be sure to be patient, make sure to record, and make sure to swirl after each drop. After about 75 drops, the pinkish color was obtained, showing that the titration of our baseline was complete.
For Part C, we wanted to determine the rate of the spontaneous conversion of hydrogen peroxide to water and oxygen gas in an uncatalyzed reaction. We put some hydrogen peroxide in a beaker and stored it overnight. After that, we added 1 milliliter of water and then 10 milliliters of sulfuric acid using extreme care when handling and pouring. After mixing that solution, we removed a 5 milliliter sample and put it in another beaker. After that, we added some potassium permanganate 1 drop at a time, gently swirling the solution until the solution maintained its color, either a brown or a pink. Then, we checked the amount of potassium permanganate we used and recorded it in our data table. We discovered that the amount of hydrogen peroxide spontaneously decomposed was 3 milliliters, and the percentage of hydrogen peroxide that was spontaneously decomposed in 24 hours was 14.29%. This means that since the hydrogen peroxide was left overnight, the rate of the uncatalyzed reaction was 14.29%. We can then use that percentage to compare it to the reaction rate of hydrogen peroxide without an enzyme to the reaction rate of hydrogen peroxide with an enzyme. The final part of our lab had the purpose of determining the rate at which a 1.5% hydrogen peroxide solution decomposes when catalyzed by the purified catalase extract.
The experiment is fashioned to test H2O2 levels after the following intervals of time: 10, 30, 60, 120, 180, and 360 seconds respectively for chemical reactions to work in each cup. In this way, we were able to test the effect that time had on enzymatic reactions and subsequent substrate levels. As for the procedure, first we placed 10 milliliters of 1.5% molar H2O2 into 6 time-labeled cups. We then proceeded to add 1 milliliter of catalase extract. To increase productivity and allow for a more controlled experiment, we decided to begin testing each cup simultaneously. For each cup, we then swirled for 10 seconds before adding 10 milliliters of H2SO4. To analyze the effect that time had on enzymatic activity, we used a method of titration, a method used to determine the unknown concentration of a solution. In this case, we assayed for the amount of H2O2 after each in time interval, separating 5 milliliters of each solution, adding drops of potassium permanganate, until the color of each solution turned to a persistent brown or pink color.
To tie our data together, we compared the amount of H2O2 used in each cup to draw our conclusions. With a baseline of 3.5 milliliters of potassium permanganate established earlier in the lab, we were able to see how our new solutions compared in hydrogen peroxide concentration. The most KMNO4 was needed at the 10-second interval because of the higher levels of hydrogen peroxide still remaining. By 360 seconds, the most H2O2 substrate had been broken down into water and oxygen, requiring little titration. These findings led us to conclude that as more time passed, H2O2 was consumed at a lower rate. However, more was consumed in total. The enzymatic activity of catalase had the highest rate of reaction in the shorter time intervals, and declined greatly towards the longer times. This lab opened our eyes to the importance of catalase in living organisms. Many living organisms require the respiration of oxygen, which in turn donates electrons to the formation of harmful substances like hydrogen peroxide. Without catalase, toxic substances would not be eradicated from our cells and would cause lasting or even deadly damage to organisms and their DNA.
This graph illustrates the data we collected throughout our lab. As you can see, the more time that passed, the more H2O2 was catalyzed by the catalase. Our data concluded that we can calculate the rate of a reaction by measuring over time either the disappearance of a substrate, catalase, in our lab, or the appearance of our product, as seen in our graph. To further our investigation, we could determine the effect of temperature or pH on the amount of product produced in relation to time.